Okay, so I'm going to change tack a little bit and talk about transcriptional targeting in order to mediate regulation of gene expression in particular cell types. So just a little bit about Synpromics. We're a biotech company located in Edinburgh, Scotland. And our particular area of expertise is in the control and regulation of gene expression, specifically to enable conditional and cell type specific regulation of gene expression. We've now grown to approximately 20 scientists in the team, all focused on creating synthetic promoters for cell and gene therapy applications. So I would say we're probably one of the biggest teams internationally just devoted to promoter development. We've recently developed a database of information on transcription factors, on different aspects or different enhancer elements that regulate gene expression. We've termed that database PROMPT for Promoter Precision Technology, and it allows us to identify and rank enhancer elements that we can then use and incorporate into our promoter design process. We validated our technology in a number of different projects, both in terms of cell and gene therapy and also in terms of bioprocessing and industrial biotech and even agricultural applications earlier on in the company's history. We've got multiple partnerships with leading companies in the gene therapy field, some of which I'll show later. And particularly we're interested in creating promoters that have very defined characteristics in terms of their ability to regulate different levels of gene expression, their ability to regulate targeted gene expression, introduce concepts or introduce capabilities such as inducibility or responsiveness to environmental or pathological conditions. And of course, because we're developing novel promoters with sequences that don't exist in nature, then we've got a very robust patent portfolio in terms of both the PROMPT database that we've developed that allows us to identify elements that we include in promoter design, and also for our partners, the sequences themselves that come out of our process, which are unique sequences and don't exist in nature, and have the ability to patent those for our licensee. So over the last year or so, we've been focusing mainly on cell and gene therapy applications of our technology. Perhaps the first project that we did was also with uniQure, so developing promoters for liver. We've also have a collaboration with a leading pharma company in developing promoters for CHO cell bioprocessing. And more recently, we've established collaborations with Adverum AJTC and extended our collaboration with uniQure, particularly developing for Adverum ophthalmology-based promoters, and for AJTC we're working on multiple different targets. And the extension of our collaboration with uniQure is actually employing our screening platform in order to test activity of some of the promoter candidates that we've developed in vivo. And then in the cell and therapy space, we're working with the cell and gene therapy catapult in the UK in order to create promoters that can be better used for the production of viral vectors. So I put together this list, really a wish list in terms of what you would want for a promoter for gene therapy applications, and looked at what's currently available. And you can see from this slide that some of the advantageous features that you really require for gene and cell therapy applications are not currently available with technologies that we have to our hands traditionally. So the ability to regulate gene expression from a cell type specific fashion, or inducible, or the ability to induce suicide if the cell therapy's gone wrong. Also the ability to have multiple genes in a construct, develop small promoters for viral vectors that have limited packaging capacity, being able to choose your expression level, so titrating up or down the level of expression you need in order to mediate a therapeutic effect, and also creating disease and environmental specific promoters. So each one of those features is not, really you can't get them from currently used promoters, and that's essentially what we're able to deliver from Synpromics by looking at the genome and identifying natural enhancers that confer some of these features, and then incorporating these enhancers into the design of synthetic promoter constructs. So just a little bit about our technology. So this slide illustrates the prompt proprietary engine, where essentially what we're doing is leveraging bioinformatics and functional genomics type approaches in order to identify and rank natural enhancer elements which confer specificity to natural endogenous promoters. And then using our machine to identify what are the optimal enhancers that mediate that particular expression profile, and then incorporate that into the design of a synthetic promoter. So in order to do that, we rely heavily on bioinformatics and the construction of, or the employment of our proprietary database. We use high-throughput next-generation sequencing-based screening methodologies that can be conducted in vivo or in vitro. And then from the data sets that come out of those screens, we employ machine learning methodologies that allow us to actually rank and identify what are the best enhancers to include in promoter design. And this enables us to actually not only develop improved promoters, but also look at other aspects that regulate transcription, so introns and UTRs and what have you. So this is very much, I'd call an engineering biology approach, where we have this design, build, test, learn iterative cycle that we employ for each project that we run at Synpromics. So the first design phase is essentially the functional genomics analysis, and the meta-analysis on different data sets so we can identify what the important component parts. Then we use traditional construct assembly, DNA assembly, or DNA synthesis methodologies to create libraries, which we then screen using NGS technology in vitro or in vivo, and then using the machine learning algorithms there to identify and rank different elements so that we can include them in promoter design. Just a little bit about our database. So this slide, the top part of the slide, shows how we start off analysis. So we'd start off with functional genomics, so differential gene expression analysis on the target cell type of interest in order to identify genes that are upregulated in that condition. And then do transcription factor network analysis in order to identify the transcription factors that control that gene expression profile, and then an individual characterization of each natural promoter that results in this differential upregulation in the condition of interest. We then put that through our internal database where we're looking at DNA-protein interactions, we're looking at cell type-specific transcriptional start sites, active chromatin marks, chromatin spacing, nucleosome positioning, which we all feed into the machine learning process that allows us to rank and identify the enhancer regions. So, I mean, as you can imagine, developing technology that is able to improve upon gene regulation has vast applications amongst the biotech industry. We particularly focused in cell and gene therapy and also bioprocessing applications of our technology. And you can see as you delve deeper into cell and gene therapy, there are multiple subsectors that we're interested in developing projects in. So you've got traditional gene therapy, gene editing, immuno-oncology, regen med, and oncolytic viruses. From a bioprocessing point of view, we're interested in creating promoters that enhance better biologics manufacture, in addition to promoters that we can use to develop stable cell lines for viral vector manufacturing. So this is quite a busy slide. It shows some of the advantageous features of our technology across those multiple different applications that I just illustrated. So these can be grouped into safety, efficacy, and design. So the ability to confer cell-specific expression limits off-target effects. Ultimately, you could reduce the vector dose, and then you can develop promoters that are inducible. For efficacy, it's, as I mentioned, the ability to vary expression strength, predictable expression, the development of Boolean logic gates, endogenous promoter editing and conditional expression. And for design, we're vector agnostic. We're involved in construct optimization. And you can see that each one of those features has applications across those seven domains that we're interested in. So this is our partnering slide, so it gives an indication of how we work with our partners in terms of the partner will approach us and give us a design brief. We'd then propose a project plan, how we would use our technology, and then we'd form that experiment and work with our partner to create promoters that meet their needs. And then the partner validates those in the disease model of interest. So just to show some case studies on some of the different projects that we've been working with at Synpromics, here we show some of the promoters that we've been developing for the liver. So the task here was to create promoters that were less than 250 base pairs in size and still mediated liver-specific expression. So we used our bioinformatics methodology in order to create a library. In this case, the library was 10 to the 5 different combinations of elements that we'd identified. And in this case, we used a fax-based approach in order to screen the library. So we introduced the library upstream of GFP, then sort cells using a fax area into different populations, and then rescue the promoters from those sorted cells by a PCR-based approach, and then do individual clones from these rescued promoters, and then test the activity of each one of those promoters using a different reporter system. So in this case, it's luciferase. We're comparing to the LP1 promoter, which has been used in clinical trials for haemophilia. And you can see that depending on the cell type of interest, so we've looked at human primary hepatocytes and different cell lines like Q7 and HepG2, you see anything from about a 5 to 10 to 40-fold increase in expression over currently used promoters. And when we looked at expression in non-target cells, particularly in cells that you would use traditionally to do vector manufacturing, like AAV, for instance, 293 cells or HeLa cells, you can see that there's absolutely no activity of those promoters in the cells that you would use to manufacture virus. Another example is where we created promoters for keratinocytes, and this was for a DNA vaccine project that we were working with a company called Touchlight Genetics in London, where essentially we were creating promoters that were active in skin cells. We then tested the activity of these promoters in vivo, looking either by administering constructs through a subdermal injection or through an intramuscular injection, and then monitored the expression of our promoters either in keratinocytes in vivo or in muscle cells. And we saw pretty well targeted gene expression in vivo, and then when we isolated tissues from these animals, we saw that you only saw expression in the liver with CMV-treated animals compared to the synthetic promoters where you didn't see any off-target expression. We also used those promoters on a therapeutic model, the TC1 challenge, where we were expressing the E6, E7 tetramer derived from the HPV virus and using that as a mechanism to protect against the growth of TC1 cells in these challenged animals. And you can see that the control animals all had to be sacrificed by 21 days because of the growth of a TC1 cells which contain or are transformed with the E6 antigen from HPV, whereas promoters from the keratinocyte-specific promoters and also CMV constructs were able to protect these animals from tumor growth over a period of 100 days. Finally, in terms of bioprocessing, this is some of the results that we've had from developing promoters for CHO cells for biologics manufacturing. And you can see, again, that we have this fax-based approach in this instance where we had created a library of elements specific to CHO cells, sorted that library by fax, rescued by PCR, and then isolated a number of different promoter candidates that were anywhere up to 9-fold more active than CMV. Then we put erythropoietin downstream of those promoter constructs and then monitored secretion into the media. And again, you can see a 4 to 5-fold increase compared to the controls. And then when we create stable suspension cells from these constructs and look at expression over time from the different synthetic promoters and CHO cells, and compared to CMV, you can see CMV silenced over time, whereas we see expression from the synthetic promoters over multiple population doublings. So I'd just like to end here just by kind of highlighting some of the key people involved at Synpromics. John Brown and Sarah Haker are here at this meeting as along with me. David Venables is our CEO. He's a leading Blythe in gene therapy. He's got a lot of experience in terms of running gene therapy companies and also bringing products to market. So I'd just like to finish there, and I'll take any questions outside. Thank you very much.